So in the previous video we left off from the indexing section, but I forgot to discuss about the top hat. Top hat is also a tool that we need for the alignment part. It's a junction mapper for the RNA-Seq reads. Like the DNA-Seq reads, there are also RNA-Seq reads that we need. It usually top hat is used to align the mammalian-sized genome using the high throughput short read aligner bow tie. So it's built on bow tie, but it works on RNA-Seq reads. It analyzing the mapping result to identify the splice junction between the exons. I hope you understand that. So in the last lecture, we created a SAM file. What basically SAM is? SAM file is a sequence alignment map format, which is a generic format for storing the large nucleotide sequence alignments. SAM aim to be a format that is flexible enough to store all the alignment information generated by various alignment programs, and also is simple enough to easily generate by alignment program and converted into existing alignment formats. And it's compact in file size. It takes less size. It allows most of the operation of the alignment to work on stream without loading the whole alignment into memory. For example, it allows the file to be indexed by genomic position to efficiently retrieve all the aligning process to a locus. It also aims to keep that file in size. It also provide various utilities for manipulating alignment. So these kind of stuff are happening with the SAM file. So SAM tools are the tools that provide the various utilities for manipulating alignments into different other types of alignment format. For example, BAM format. BAM is the SAM is human readable format. The humans can read that format and can understand that. But BAM is the binary alignment map. So BAM doesn't contain that kind of information. For example, if you create a BAM file, you can't visualize it. You can't see it. There is also programming language and something like that. So BAM file is just like that. So for converting the and sorting the SAM file, we need SAM tools. So first we are going to install the SAM tools, and then we are going to convert the SAM file into BAM file, and then we are going to sort that BAM and SAM file. So let's just do that. So copy this command into our text file that we usually do in our part. So here we are going to install SAM tool, which is already done in my case. I'm just gonna clear and see that I'm in the right folder. Yes, I am. So I'm just gonna keep it there, and there is a SAM file to a BAM file converting pattern that is present here. So gonna copy this command and paste it in our terminal. So SAM tools view, yep, align.sam to change into align.bam. So it has done. There is should be a file, yep, align.bam file is here, align.sam file is here. So we are going to sort the SAM and BAM file together. Sort.bam and align.bam. So I think there should be a mistake that I'm seeing sort.bam file to be created from align.bam file. So sort.bam, I think there should be a problem with this. Yep, there was a problem. There should be a file now, sort cat sort.bam. Yes, so here it is. So cat sort.bam file. It now you can see why I called it not human readable. So it is a machine readable file. It's a binary alignment file. So that was the file that we used. For example, if we took our SAM file, we can't use the SAM file to just manipulate that file into different different other steps that we need in the further process. So we need a binary file. We need a binary alignment file that we could use, that machine could learn from, that machine could use. So we created that file. We changed our SAM file into a PAM file, and then we sorted that PAM file using the SAM tools. For if you want to play with your BAM files, there is also a utility that is present there, which is called BAM tools. You can use BAM tools. If I can see, I have BAM tools install. I should have, yet I got the BAM tools here as well. I can use that BAM tools to convert between the BAM and number of other formats. I can use the count, I can use the header, index, and a lot of other stuff that I can use with the SAM file. I can use with the BAM file as well. So that was it for this lecture. So we have successfully, we are, I think, at the end of this lecture. Just keep in mind that we are using our demo file, which contain only 2 reads. So NGS data sequencing is not this fast. It is much faster. I'm just typing the command and then the command is getting done. We are just now need a reference annotation file, and then we going to cut out our annotations from that annotation genome files. So that was it for this lecture. In the next lecture, we are going to download a GTF file of our genome. I also gonna show you how to download your GTF files. Then we are going to extract our RNA expressions, which is also the DEGs, differentially expressed genes. We are also going to analyze that genes, and we are also going to make a heat map of it. So which you already did in your last session, I think, which is not, I think, is required in this section as well. So in the next lecture should be the last session of our command line section. So that was it for this section. Thank you.